Alright guys, welcome back to Underground Science, and in this video we'll be quickly going over a basic overview of B lymphocytes. Alright, so let's go and get started. So to start off with, if you guys remember, the B lymphocytes, right, were part of our adaptive immune system. So let's go and write that down. So the B lymphocytes were part of our adaptive immune system, also known as our specific part of our immune system. All right, so also known as also known as our specific part of our immune system. So remember, we had our innate or non-specific part, and then we have our adaptive immune system, and then specific. All right, and also the B lymphocytes so it's part of our adaptive immune system, right? Our specific adaptive immune system, but it's also part of our humoral response in our adaptive immune system. So it's also part of our humoral response. All right, so that's that. All right, so now let's just go and look at an overview of B lymphocytes real quick. So let's say we draw a B lymphocyte like so, we'll just draw like a circle. All right, so or maybe someone like a circle. All right, so let's say this is a B lymphocyte, even though it might look like a macrophage or something, but it's fine. All right, so let's just label this as a B lymphocyte. Okay, and it turns out that on these B lymphocytes, we have around 9 to 10,000 membrane-bound antibodies. So immunoglobulins, or immunoglobulins, couldn't speak there. So it turns out, so let's go and draw some. So as we know in our antibodies, right, we have some heavy chains and light chains that make up our antibodies. And let's go and draw a couple here, or actually more than a couple, right? At least try to with this amazing artwork that I have. So, kidding, no, that's horrible artwork. But yeah, so let's just go and draw like one more. And there you go. So these are our membrane bound antibodies. All right. So these are membrane bound antibodies that are part of our B lymphocyte. All right. And it turns out that we have around, well, we can give it a bigger range if we wanted to, right? Let's just say it's around 9 to 11,000, all right? Or 9 to 10,500 will be, I guess, a little more accurate. So our B lymphocytes can have around 9 to 10,500 membrane-bound antibodies, right, on its cell membrane. So that's why it's called membrane-bound. All right, so that's a lot. And so what the job of these antibodies are is to actually bind to specific viruses or antigens, right? So if we have like a bacteria floating around here, right? Like let's say we have a bacteria that's floating around here. So let me go and choose an orange color, I guess. So if we had a bacteria here, right, that was floating around, right, trying to do some damage, if it runs by our B lymphocyte, right, our B lymphocyte would go beast mode and try to attach. So let's just show our bacteria floating around here. Our B lymphocytes are there right near the bacteria, and then eventually, right, our B lymphocytes would actually try to join to the bacteria using its membrane-bound antibodies. Or it's not, never mind, it's not happy, right? So it would try to join and bind to the bacteria, all right? So it would try to bind to the bacteria, and the spot that it binds, right, this spot right here, is called the epitope, all right? So the binding spot between the membrane-bound antibody on the B lymphocyte and the antigen, or the bacteria in this case, the binding spot is called the epitope. All right, so let's go ahead and write this down. This orange person right here that's really mad is the bacteria. All right, it could be a bacteria or a virus, right, or any pathogen that runs into our B lymphocytes and our antibodies. And by the way, it's not just going to bind to this bacteria, right, just like randomly, because remember, this is part of our specific slash adaptive immune system. So it's only going to bind if this bacteria is like, if the binding spot of the antibody surface, if the variable portions of the antibody and the bacteria match, if it matches and if it can successfully bind, it'll bind. However, if this bacteria, like if there was another, let's say a virus floating around here, and the virus was like this, right? So if we had a virus like right here, so let's go and label this as a random virus that's floating around. Just because it's floating around and it's a pathogen doesn't mean that this specific B lymphocyte will have the specific spot on the surface of their antibodies to bind here on this virus. Maybe, so let's say we can draw something like this. Maybe it comes across these membrane-bound antibodies, but it won't bind. So maybe this virus floats around, right? Somehow comes here. However, maybe the specific variable portions on the surface of the B lymphocytes. So maybe the B lymphocytes, the binding spots of our surface of our B lymphocytes, or sorry, more specifically, I should say, the surface of our membrane-bound antibodies on our B lymphocytes, maybe they might not be applicable to the virus. So maybe the binding spot won't be able to, like, the binding spot on the virus just maybe won't match, right? So it doesn't mean that just because there's a random virus that this B lymphocyte will inhibit it from functioning and make the membrane-bound antibodies bind to it. There might be another B lymphocyte that matches, right, that these variable portions might match to the binding spot of the virus on another B lymphocyte. But this B lymphocyte, it might not match, right? Or it might match, but with this bacteria, right? So things like that. It doesn't have to match all the time. And know that all of these antibodies, right, on the B lymphocytes membrane all have the same binding spots, all right? Because recall, they also have DNA, right? Because they have to replicate into more B lymphocytes. For example, right now, if once we have a bacteria that binds, right? Usually we're going to talk later about how we need T cells most of the time, right? But if we go ahead and ignore that for now, right? We know that eventually, after the B lymphocyte has a specific pathogen or something that has successfully attached itself on the surface of this membrane-bound antibody, the B lymphocyte can then undergo mitosis, and it can actually start replicating itself into two things. The B lymphocyte can replicate itself into memory B cells, so memory B cells, or memory B lymphocytes, whichever one, right? And it can replicate itself into plasma B cells, so plasma B cells, all right? And another cool thing to know is that another word for plasma B cells are B effector cells, all right? So B effector cells. And so, yeah, so these are the two main things that the B lymphocyte will start replicating into, and very fast, as the second we have some pathogen that has successfully attached itself onto the variable portions of one of its membrane-bound antibodies. And so it's only fair to say, if we take an example of a memory B cell, right, it's important that it replicates into memory B cells because, like the name, it's going to kind of remember, right, this bacteria, this specific bacteria. So next time this bacteria or some type of pathogen comes along, we have many more ready, and it'll decrease the time it takes to get that bacteria or pathogen out, right, to get it taken care of. But plasma B cells are pretty much B cells that produce a bunch of antibodies. All right, so they pretty much are B cells that are responsible for producing antibodies. All right, and these antibodies can then go and help the site of infection, or it can even float in the blood, obviously, and tag viruses and pathogens there, right? And these antibodies, by the way, right? These antibodies are going to be the same as the antibodies on the surface of the original B lymphocyte, because remember, the trigger originally was the bacteria. So we want the same exact binding portion on these antibodies, so we can take these antibodies and go find somewhere else where this bacteria might be sneaking around, so we can destroy it. All right. So, and memory B cells are good for like reintroduction of the same bacteria later on. We would have memory B cells that will remember this bacteria and attack it quicker. All right. So the response, the immune response, would be faster. Of course, we need T cells, but if we just take the picture of T cells out for now, we know eventually, right, with the help of T cells and the binding of some pathogen on the membrane-bound antibodies of the B lymphocyte, we're going to see mitosis of the B lymphocytes and a lot of proliferation into memory B cells and plasma B cells, which are pretty much B cells that produce a bunch of antibodies. Another cool fact is that the plasma B cells can produce around 1,500, oops, did I make a random dot somewhere? Sorry about that. Can produce around 1,500 to 2,500 antibodies per second. All right. So it goes insane mode. All right. So produces 1,500 to 2,500 antibodies per second. The plasma cells do. All right. And once these antibodies are free, right? Once they're produced, they can go ahead and find this particular binding site on this pathogen, right, or this antigen, and go and bind to it, right? So find other similar antigens with a similar binding site, right? So let's say it's like this. You can bind to it, and then it can tag it, right? Or you could even, since it has two parts to it, right? The antibody does. So what it could do is it can also bind two at a time, also, even if it wants to, right? And what this does, it actually inhibits the pathogen from functioning, right? It's kind of like tagging, and the process of the antibodies tagging these viruses or bacteria, whatever pathogen or antigen it is, the process of these antibodies doing that, right? Tagging it for pickup or tagging it for other white blood cells to come and take care of it, right, is called opsonization. All right, so opsonization. All right, and so this will allow things like specific types of white blood cells, like it could be, let's go list them out. So right here, it could, so the process of opsonization could attract specific white blood cells like macrophages, right, like macrophages, neutrophils, and maybe even dendritic cells, right, over to this area of where these antibodies have opsonized and have attached itself, right, to that specific pathogen before this B cell started replicating itself. So, and remember that these are types of phagocytes that we have in our immune system. All right. So that's pretty much it. That's the basic idea that I wanted to get through in this video, right? And also, right? Sorry about that. And also, B lymphocytes, right? So if we talk about where they're made, right? So all blood cells, right? Most generally, all blood cells of our body are made in the bone marrow, right? They're made in the bone marrow. However, they also stay in the bone marrow and actually mature there. So what I'm trying to say is that the bone marrow is the site of maturation for the B lymphocytes. So this is where they mature and divide and grow. And we'll see that it's different for T lymphocytes. T lymphocytes start getting made in the bone marrow. However, they go to another a lymphoid tissue spot, right, in the body where they mature. All right, but we'll look at T lymphocytes later. All right. So that's pretty much it. That's all I wanted to get through in this video. I hope this video made sense to you guys. I hope to see you guys in my future videos. We'll talk about more cool topics about biochem, physics, and chem. Don't forget to hit that notification bell, like, and subscribe. See you guys later and stay safe.